So now it's my great pleasure to introduce Dr. Suma Jacob, who is here for a distinguished lecture. She is a distinguished child and adolescent child psychiatrist, and as most of you know, the new chair of the Child Division of Psychiatry. So, yeah. So, and we're super happy to have her. I'll tell you a little bit about her background, which I'm sure you can find on the internet. So I'll keep it quick so that we give her enough time to talk. But, she really works, she's an MD PhD, and she was an undergrad and did her MD PhD at University of Chicago. She had a bachelor's in psychology with honors, of course, and a thesis. And then she did an MD PhD at University of Chicago, which we all know is a place with huge grade inflation and where people coast and don't work that hard. Not, okay. So incredibly impressive, background through her training. And then subsequent to that, general psychiatry and a fellow in child psychiatry at University of Chicago. And she has so many awards, like her I just have the first 5 pages of her, CV, and it is very, very thick. But I just pulled a few out of here that I thought were fairly remarkable. She was a Kavli Fellow at the U.S. National Academy of Science, France U.S. Frontiers Program. She's a full member of Sigma Chi Scientific Research Society. She's an awarded associate member of the American College of Neuropsychopharmacology, and there's been the Laughlin Fellow, the American College of Psychiatry. She's won numerous mentoring awards as well, and I'm sure she's going to be, aside from her research and leadership, an extraordinary mentor to our future and trainees. And so we're super excited. I mean, I would take her lecture to literally read the honors and awards, so I won't. But her work is in social neuroscience, neurobiology, and neurodevelopmental disorders across the lifespan. She's done everything from kind of clinical trials and biomarker and even genetic studies on autism, and has a focus on novel phenotyping methods related to neurodevelopmental disorders, including OCD and tic disorders, anxiety, ADHD. At University of Minnesota, she was the Gloria Siegel Family Endowed Professor and the associate director of the medical scientist training program. So she's been involved in training MD PhDs in a leadership role. She also served, which I did not know, as the chief medical and science officer for a large nonprofit community mental health organization that was focused on innovative integrated health implementation, something that is hugely needed. If I can take a quick diversion, I've heard psychiatry and mental health in the United States being described as a system that was designed by 10,000 architects who never talked to each other. And so there's stairways leading to nowhere, doors that don't open into anything. It's just a total, mess of disconnection. And, there are very few other areas of health that really look like that. So this kind of thing is widely needed. I look forward to working with her, over the next decade and more to fix some of those things. So anyway, thanks for coming to share some of your scholarship with us today, Suma.
Thank you, everyone, for inviting me. I'm so excited. Some of you know I trained at UCLA in the early 2000s, and coming back has been really exciting. And then giving this talk, I'm so excited because there are mentors in the audience that I've known from undergrad days, and just the span and breadth of autism work at UCLA, everything from genetics to imaging and school-based work and incredible phenotyping and clinical work. I know few other places that have that span. So today I'm going to talk about something very challenging in our field, which is the diversity and heterogeneity of autism. And I'm going to change in the order I talk about phenotyping last in the talk, because that's where some of the innovative work is. But I also want to really hold it in context of child psychiatry and transdiagnostic trajectories, because autism overlaps with a lot of different things. So there's multiple levels of heterogeneity. I'm thinking about people in the audience who've done genetics and more molecular work. It's so complex at that level, but then you go to intermediate phenotypes, and there's another level, and then you go to clinical phenotypes, and then you think about currently the social definitions and terms of what we consider autism. So it's a daunting task when you look at all these levels. I'm going to talk very briefly about one of the largest clinical trials just to illustrate the challenges of then translating what we know in biology to a disorder / condition as autism. And then I'm also going to talk a little bit when I talk about stakeholders and partnerships, which have made the whole process of who we're serving incredibly complex to think about it thoughtfully as a group. So as Dan mentioned, I did MD PhD training, and the dream of that approach was that you would take bench to bedside. And some of the challenges we run into that is there are a lot of intermediate steps, and least of which Dan mentioned, like how we get integrated healthcare to individuals when they have to go do things piecemeal. There's multiple different contributions to disorders. When I started studying this, I remember thinking about Fragile X, where we know that there's a gene, but even with that, there are very complex presentations. And that variability goes throughout when you think about all these different things. So in child psychiatry and child neurology and many of the fields where developmental pediatrics, where people see kids with autism, there are so many things that have to be addressed. So intellectual disability, communication level, motor, tic disorder, as well as many medical and other comorbidities. And I'm a neurodevelopmentalist. I like to say that you can't, a lot of things were defined by adult disorders and brought back to child. And if you start at sort of the fetus and young baby and go forward, things would look very different. And so even the idea of having very narrow diagnoses and studying them, which is what we try to do as scientists to control variables, doesn't help us translate it back to the patients we see. So that's a huge challenge. And many of you have seen this image before, but with autism and the core symptoms of restricted repetitive behaviors and social communication, we've gone through like 2 decades where we've really looked at genetics and contributing causal factors, looked for biomarkers, have gone up and down with some things that work, some things that don't. It's been extremely challenging. Medical comorbidities like seizures and GI and other effects, and then this whole range of behavioral and psychiatric comorbidities. I'm putting in weight and sleep in there because this kind of summarizes what we have for treatments. So currently no medications for core symptoms, but there are incredible, well-studied psychosocial and educational interventions, many of whose experts are in this room. And, but that continues to be a big piece of what are the range of biomedical services we offer, as well as social cognitive services. There are communication tools for speech and language, but a lot of traditional speech and language don't serve this community as well as they could. ADHD management, you will see in the literature everything from 30% to 80% have comorbid ADHD. And there are medications that have been studied in autism that work, but that's a unique, challenging combination. And I will address this because if you offer a child social skills training or something else, but they can't attend to what they're learning, then you're not setting them up in the ideal situation. The only approved FDA meds are risperidone and aripiprazole, and they're to treat aggression and irritability. And there are psychotherapies out there. CBT for anxiety is actually what has the most evidence, but mood and disruptive behaviors, there's still a lot of challenges. There's newer research that metformin can be used for metabolic weight side effects, especially when you have to use some of the meds that cause that weight gain. And then there's the newest data on sleep says that the prolonged release melatonin is an option, but sleep is a huge issue. So just the complexity of who we're serving. I'm not going to go into the background. There's a really fun paper that I just published that Sue Carter invited me to do, which it talks about it's a bio narrative. So I talk about my PhD training, but very much in the lens of how I studied oxytocin and vasopressin, and mention how that journey went and how it affected what I studied. So I was asked to be part of one of the largest oxytocin vasopressin not oxytocin trial, vasopressin trials, which was supported by Roche. So it was an industry partnership. So I'm going to talk about this because it was published as a trial that didn't have the expected outcome, but it highlights many of the challenges we have with clinical trials. And so why vasopressin? There's animal data, some of the most, recently Larry Young passed away, but he did work on paternal behaviors in voles with vasopressin. And this particular medication had some promising initial results in preclinical trials. I was involved with all three of these studies. The very first one, and again, when we often do clinical trials, we start with adults because we are very careful with doing any sort of trials in kids. And then the next one was a downward extension into kids. And the last one was adults. And the big challenges it illustrates, the first one used SRS, which is social responsiveness scale, as an outcome measure that didn't work. And then reanalysis of Vineland, which is a developmental scale. They used components of that for social and communication. And the attempts to bring this down to the youngest age, our outcome measures have to go across developmental ages were a challenge. And so I'm going to talk about this last study, which was done across Europe and the US. I was a site, and UCLA was also a site, but it just illustrates some of the challenges of coming up with new medications to target core symptoms. So again, they tried to limit higher IQ in the sense of above 70, certain levels of severity of symptoms, and then from the earlier trials settled on a dose, and outcome measures were at 12 weeks and 24. And then a lot of these initial trials, is it safe? Is it going to work without harm? And then we'll look at the outcome measure that was selected. There wasn't a good enough outcome measure. And so they went through and pulled together 9,000 Vinelands that are publicly available, came up with this composite score, this 2DC, used it as the social measure, and then put in other things like pediatric quality of life, CGI, for those of you who do trials, is sort of the standard clinician rating, and anxiety scales. And then we can look at how this varies across geographical region and age. Overall, for safety, the main point is it was fairly safe. There were no side effects. A lot of thought went into it. What was interesting to learn, the animal model had some peripheral neural stuff. So I got really good at my neuro exam peripherally for some of those things, but none of those things showed up. And then when you looked at the results, if you look, it favors placebo. And it was stopped with futility analysis after 50% of participants. Incredibly expensive study across the world. And so this Vineland 2DC at 24 weeks did not show the difference. And so let's think about why. And many of you know there's like placebo response, expectation bias, site types. So it used to be 5, maybe at most 10 sites across the country. would see all the participants in these large trials. Now there are 30, 40, 50, and you see smaller number. And so just kind of thinking about that. Recruitment, we're in this era where we recruit people on the internet and on the web versus coming to the place where they clinically saw clinicians all, and so people know the participants well. And so I'm going to talk a little bit about also baseline and outcome, and later talk about an oxytocin trial that I was involved in to highlight these differences. So baseline characteristics, where you start makes a huge difference with any trial. And this was an issue specifically, and we can retrospectively go back and analyze where everyone started. If you had a lower Vineland 2DC score at baseline, there was a greater potential to change. And so those people that came in with too high of a score aren't going to change. So that's a big issue. There are very limited pool of outcome measures, and there's limited funding to develop these. They're reliant on caregiver support people to also tell you, as well as clinician report. And then placebo response can vary depending on the outcome measure and who's reporting. So if you look at all three trials, you can see that if you use that same outcome measure, that first one that looks so promising, look at that huge standard deviation with adults in that. And then with the pediatric one, it's right in the middle. And then the replicating adult one, it's way off to the left. So this is a huge challenge in population about which outcome measure you're going to choose for which age. Many of you know that in pediatrics and then even in many adult disorders, placebo is a component that we have to deal with. And so this study also allowed us to go back and look at where was the greatest placebo response. And as you could expect, where people were recruited, how they were recruited, and even some of their baseline measures affected this. So if there are currently a lot of CROs or private clinical trial organizations, in this study, academic sites did better. They had lower placebo response. Similarly, if you were recruited from the web versus more personally recruited via advertisement or clinic, that group, the clinical group, had a lower placebo effect. And there was a lot of effort to put placebo training and management, and that still didn't, delete this huge effect. So another approach is taking these things and doing computational math and data-driven approaches. And I'll talk a lot about that in my talk, because now that we're having bigger sample sizes, we can do this. So Russell Taube and colleagues went and did this specific lasso regression, linear regression across all three trials. And what's interesting is when they found what causes increased and decreased placebo, it does come up this higher baseline, decreased placebo. So if you remember, that's going in the opposite effect. So that's a challenge right there that shows your murky results. And then the longer duration of the trial decreased placebo. The European sites did better than us in the US. So that's also an interesting contrast. Increased placebo commercial sites, and because it spanned kids to adults, the ADHD in the kids created a huge amount of variability. And then depression in the adults with autism created a huge amount of variability. Yes, Dan?
Was this done in a discovery and then a test set, or was it just done in a leave something out?
It was a discovery and then a test set.
And then a test. So all these predictors are in the test set.
Yes.
Yeah.
Yep.
Thank you.
So the other thing is, towards the end, that third trial, the belavaeptin got FDA breakthrough status. So all of a sudden, especially in rare disorders and disorders like Fragile X, parents hear about it, they get excited about it, the community gets excited, and that introduces an additional bias, expectation bias. So all of these things have to go into thinking about how we design trials. And it just tells you, as we get larger sample sizes, it's not enough to address heterogeneity. We have to consider things like recruitment sites, outcome measures, And then a lot of the trials we've done sometimes have an improvement in quality of life. And I think that that's an interesting piece because it might tell us we're not measuring the right thing that's changing. So some of our measures try to be very specific for target engagement, and this PEDS quality of life is very general. And sometimes if you have something that's moving that, but not moving the other things, you may have the wrong thing you're following. So we're gonna look into what we can do in the future. What are targetable symptoms across disorders? You can't ignore comorbidities and just say it's not there if you want to translate it back, but it introduces a huge amount of variability. Floor and ceiling effects with baseline measures. I'll talk a little bit about examples of new designs. And then this placebo and expectation bias is huge in our field. We have to come up with things better than rating scales. Rating scales are incredibly useful because the clinician in an outpatient office can use it. But many of us who've done trials, you get the father or the mother to give you the rating scale and you get totally different results. And a lot of studies don't even control for which parent it's coming from. So you have all this up and down. So we have to deal with that and develop measures that will work better. And then also biological markers. Some of the new trials I've been involved in have EEG specific biomarkers. So they're things that should be less susceptible to placebo response. And I'll talk a little bit, I've been working a lot on task-based digital markers and the ease of scaling some of those. So adaptive designs for clinical trial have become really popular, and it's sort of the future direction. And even you could see this last trial I talked about stopped at interim analysis. But this is an approach. The critique of the approach is it's hard to have a lot of sites make those decisions and have it move fast. There was a recent paper that looked at hypoglycemia related strokes, and they actually did one trial in the traditional, where they call it frequentist, and one with this Bayesian approach. And they found that when they did the exact same trial in the same population, this adaptive design allowed them to reach the same conclusion with fewer participants faster. So I do think it's a wave of the future, but I think it takes a whole level of implementing that we've yet to figure out how to do. And so a little bit on who we're working with, because this is a huge complexity. This last study I talked about started with adults. It was a first adult trial really done at that scale. What do you do for your measures when most of our measures were with parents or caregivers earlier, right? And so it had to figure those out. And the whole difference of the work I've done in Minnesota, we realized we had this newsletter that went out to the community, and over time got feedback. We may need to have a different newsletter for adults versus families and at different ages because their interests and focus were very different. So who are you serving? And because autism is so expansive, you really need to look at all these different elements of who's involved and where you're getting the data from. And it just shows you that level of complexity. What and how, because we span everything from biomedical and genetics all the way to social interventions. Where it's done. As we've advanced to do online trials, we can do things more, as many of you clinical practitioners over the pandemic saw patients like teens laying in bed talking to me. We can do so many things, but what's the difference between that data versus when they're in front of you? And then community work and online work, these are huge spans. And then even with kids, if you catch them at certain times, the summer, end of the school year, fall, your data is going to look different. And so some of these 24-week trials span that time, and we're not capturing all of that. And then why? This is a huge issue that the community cares a lot about, from everything to being sensitive about the term, like curing. Many meds used to do that, medications, and that was a goal, to who wants it. Is it the parent? Is it the child? Is it out of needing resources because they can't get an assessment? It's a very complicated process. So Roche partnered with this group with stakeholders and came up with this participant-friendly clinical trials manual. And what I like about it is it was written for why participate in trials. And many of you are aware that the community is very skeptical of what science can do for them. And what they did was come up with this targeted towards them to think about why do it. And how do we approach this? They describe an autistic person's journey or their supporter journey. And this one, there's one for different ages. This is birth through 12, but it goes through from their perspective. So as researchers, we can think about what it looks like for them when they're coming in to participate in these studies we're offering. And then it also has these very nice videos that participants can view to know what questions to ask, or families can know what questions to ask. Transdiagnostic trajectories, I'd mentioned, this is a huge piece of thinking of a lifespan disorder or condition. When and where we intervene or offer things. And if you look at child psychiatry disorders, things emerge at very specific time periods or time phases, sensitive periods. And one of the things with my background in behavioral neuroendocrinology, I've learned a ton by the difference of pre-puberty analysis versus post, which happens. There are even differences in platelet serotonin levels before puberty and after that we ignore when we just gloss over and categorize kids across it. And something like puberty is really sensitive to measure. So how do we do that well? And it expands and it changes over time. So it's, again, a classic example of the complexity of biology with these disorders. I bring this up because I've been interested, aside from puberty, adrenarche. There's some work that says adrenarche and that first release of hormones with the tic disorders and even ADHD and some of those other things could be correlated. And they're all things that we've known for decades, but are really hard to measure. So when I think about disorders or any psychiatric symptoms or presentations, I carefully think about when they come on. And one of the things that we need to think about is what we're measuring when. And so if you look at under 5 here, these early disorders, even anxiety presents in different ways, from separation anxiety to selective mutism, social phobia, and generalized anxiety. And so many kids that have one may have had others and continue to have elements of others. So this is, again, just to emphasize we can't look at one thing. And when they present and how they overlap is really important. And then as we go to adulthood, I always like the work I do in OCD, what people are compulsively focused or kind of stuck on changes as they go through this developmental trajectory. So from, the fear of contamination or getting sick to even more complicated behaviors of relationship obsessions and all of those kind of things happen over time. So we have to hold all of this in developmental context. So, this idea of how do we do measurement with this incredible complexity? There's this idea of precision mental health, but where are we? How do we optimize these sensitive periods? And then can we use these computational models where we have more data to do test samples and retests to advance our approaches? And so this idea that's been around for, a period of time with doing, thinking about precision medicine, so many of the things in medicine is one size fits all. So if just as vitamin D became a very popular thing. We had one measure that doctors say that are in this range, it didn't show the complexity of where your ancestry was and maybe what you were supposed to, it didn't address sex differences. It didn't address any age differences. So we're coming from this era of one size fits all to more the stratified. We do look at subgroups, and we're starting to use genomics and other things to look at subgroups, but we're still far from how we bring it down to that one individual person to really tailor it to that specific individual. And then with development, when we think about what are the risk factors at different time points, when things start, how they start, you're going to also have a different set of biological factors influence all of those. So when you decide to focus in, a lot of work in autism has really looked at early childhood because of the promise of shifting these trajectories. But just think about all that's happening between 0 to 5 and what you can do. And one of the studies that I've been involved with is this larger phenotype study where we did it with a community sample instead of a clinical sample and used these measures online to then bring in a subset that would get get neuroimaging or other more in person. And then how do we do it? And one of the unique things we did with this particular NIH funded grant was different computational ways of addressing the phenotype. So this is work I did in collaboration with Institute of Child Development, Jetty Ellison and Jason Wolf. And this community sample, Minnesota has in the Twin Cities, all the kids, when they're born, parents have the ability to opt in or out of wanting to be called back for research. So it's this idea to get all these measures across all the kids. And we ended up doing more traditional analysis, cluster-based analysis, and partnered with people who do anomaly detection. So especially with those people who do things like rare variants or syndromic kind of disorders, it's not just looking at averages. The kids that really stick out early, how do they look different? And so this project is still underway. One of the unique things about it with computational is you can have the data tell you what's going on, or you can do it with design longitudinally and have the outcome then re-influence how you look at the data, so supervised and unsupervised. So we're in the process of doing the longitudinal sample to look at what our predictors and outcomes are. And this is already a paper where Angela Sang, who's in my lab, along with someone in speech and language, just looked at, in this community sample, what we can learn from words produced versus words understood versus gestures. And most kids fall into their high cluster of language abilities and communication abilities. And then there's the other subset, especially when you go to early, this is 18-month-olds, that are low on everything. But the interesting group for us are these groups in the middle. And the difference between cluster 2 and cluster 3, or cluster 2 are the words that had the low words understood. And if you have these kind of markers, you could follow those kids and look at what happens. But now with computational data, we can track it in a way that we haven't been able to. And again, as I mentioned before, if the parents aren't really attuned, or you get dad or mom who's going to give you different data, it's not going to work. Ultimately, we need to pair it with technology that can actually record words and exchanges and all of these things. So it's sort of a future approach to catch things very early. The other window is teens, and even early in the early teen years, where there's plasticity in learning. And in other disorders like mood disorders, you can come up with the CBT program for adults, extend it down, and it works on young kids, but it absolutely does not work on teens because they're evolutionarily geared up for something very different, high exploration phase. And that high exploration phase is another window of interventions. And so when Sofia Vinogrado came to Minnesota, she had been working doing cognitive training with schizophrenia and had been partnering with Mersner and Posit and had these interesting results that these very focused kind of training games could make a difference in individuals that had chronic schizophrenia. So I said, if that works for that group, why not try it for individuals with autism that in an age that there's more plasticity? So these are this is our, you know, pilot data in the sense of the sample sizes of the groups are around 20 each. But I reached out to Liz Lageson, who allowed us to put this game with peers. So that I think a lot of the things we do, we don't think about social context and are they having the opportunity to learn? And what was interesting about this again is this quality of life improved. And if you look at the bar on the lower left, with the cicadas training and peers, they looked better than either of those alone. But the task outcome measure was very specific, didn't work with the game training alone. And when you look through the games, what's interesting, there's very simple ones like sound sweeps. So sounds going up, sounds going down, can you detect the difference? Similar to schizophrenia, people, when they train on these things, it makes some sort of difference when they're in social contexts. And generally, maybe they're attending to things that they normally don't because they're really focused on visual. And the In the Know is this really fun cartoon game of you're in these social situations in the break room at work, what do you do? That was something that really moved in this training paradigm. So this is something I'm hoping to follow up. There's a lot of overpromise with digital and, these kind of things, but I think they don't often look at the context that you actually get to practice what you learn. I'm going to talk very briefly about the oxytocin trial that I did, but in the context of we can't do clinical trials without knowing whatever the compound was for the history of biology. And so I always say with these neurohormone trials, would we just give someone insulin once and say it's supposed to work? It doesn't make sense. So hormones are feedback regulated. They're very complicated. Doing invasive studies, especially in kids, is very challenging. I was able to partner with individuals that have these great models and resources to look at what happens to this oxytocin system. And then we did some reverse translational studies where the TD monkeys got the exact same protocol as the kids in this oxytocin trial. And then we could also follow them out of what happens when they got it when they were little with puberty, and then outcome-wise, look at things like their reproductive physiology, because there was concern that it would affect long-term reproductive physiology. So those were all really interesting things to think about. And with Evdokia Agnestou, I was able to do this adolescent-focused trial that was, again, small, but it was very focused on a task that paralleled. So you're going to say, what do faces have to do with animals? But it was more identifying what's similar or different. And with oxytocin in animal trials, they're able to tell differences and discern similarities and differences. And this trial picked that up, that in faces, you could tell similarities or differences better with oxytocin, but not with houses. And we could actually measure that the oxytocin got in the system. And most of you, and then this PEDS quality of life was better with the oxytocin versus not. So what are we measuring? Is this task a better identification than not? And so many of you heard there's a very large trial, but what's the difference between this very large trial? Age 3 to 17, huge age difference. Outcome measures were the rating scales, not tailored to what oxytocin does evolutionarily. Our study also did the same outcome measure, which was negative in both studies. So it's not picking any difference up. The study used a completely different formulation of oxytocin, so that could have an effect. And there were a lot of issues with that. And then the heterogeneity of the sample size, that study really tried to say, will it help young kids who have more severe challenges or intellectual disability? But it made it very hard to detect any subgroup of responders. And again, timing you give it. When I did this trial with Evdokia, she said, oh, let's do it, 4 hours max half-life. Let's do it at 9 and noon. And I said, in the US, there's no way I can get noon as a dosing. So let's do it in the morning and right after school. Like, we have all of these challenges of what works and when and what's optimized. So as we get to this era of big data, we have more data than ever, and we're thinking, like, how do we use this to think about clinical phenotyping and improving the studies we have? And computational psychiatry, I had the opportunity to work with David Reddish and others at Minnesota, and I love this image of this pool of water, and with causal discovery or other kind of methods, we're like, where did it come from? What did the ice cube look like? And it's a very hard, challenging thing. And so with these methods, there are these mathematical approaches. And I also like this greedy search approach. There are all these trees, there's all these details. How do we figure out algorithms? So again, this is a study that's in already preprint version that we're getting out. But we took the Simon Simplex sample, and I was talking last night to Kathy and other people. This was an old sample that was much more homogeneous in the sense of the time when it was captured than some of the current samples. And what we did is this really agnostically, to some level, look at what are the factors that look different in girls versus boys. But to do that, if you put everyone together, this causal discovery, the mathematical algorithms will look at what drives things either in positive directions or negative directions. And I just really want to highlight the quality of the data that you put in is going to drive whatever you get. So all the detail that we put into thinking about data makes a huge difference. The other thing is, I really think these methods should be used to come up with our hypotheses and then do follow-up studies. That this, like, no mathematical algorithm is going to tell us exactly what causes things, but it really gives us clarity and sort of seeing things in new ways that's data-driven. And so if you look at age, nonverbal IQ, and verbal IQ, if you put everyone together versus what we did is there was a smaller sample of girls, I think 334 girls. We sex matched, IQ matched them with boys, and then compare. So those larger samples, we looked at that combined version, and then with the general. And verbal IQ really was driven and was one of the driving factors in the girls. And this was not really picked up when you looked at everything together. And some of the directions of what you look at, verbal IQ can drive social emotional awareness and then social atypicalities. And then the verbal IQ also really affected restrictive repetitive behaviors when you looked at that differently. So these are some of the methods that we can start using to generate hypotheses as we look at these things. And then the clinical piece, does it make sense? When you create these, we had to pull out that sex doesn't drive age and age doesn't drive sex. You have to really, like, you just can't throw in all the data and not make sure it makes sense. And then you can also do things like Dan mentioned, test it on one sample, test it on another sample, to really see that it holds up. So there's a lot that I'm hopeful that with computational innovation, technological advancement, methodological innovation, we're going to be able to do more things. Sometimes industry is challenging, like there was this great sleep pad, because many of you know that kids with autism will tear off things that I had heard about that I wanted to use. And then the company, like, which version of the computer that it works or not fell through. And so we have all of those challenges, but hopefully we will get better data over time to do more specific effects. And I always laugh. This is like this kind of a funny story. My watch constantly measures my heart rate. And here I am every time I get an alert, even if I'm totally calm, I'll look back and I was giving a talk. Because my watch knows like that even on externally, if I'm calm, that I'm at this amped up, heightened state, that in a resting body state, I shouldn't be this amped up. So I think the future of that kind of data will be really, really helpful to think about what we do. And then this is just, labs, multiple people that have helped over the years, including Dan had mentioned the large community mental health practice that I participated with. They allowed us to do a lot of these bigger data projects and collect data. So thank you, everyone.